Hey guys, and welcome back to Now I Know. Today we are talking about cell cycle. Now when I think about it, I feel really amazed, that me, you, or any multicellular organism, especially when I talk about humans, we all are produced from one single cell. How amazing, right? This one single cell has given rise to me or to you, any multicellular organism, right? And that is because the cell undergoes cell cycle. Okay, now in human body there are trillions of cells. This one single cell will give rise to trillions of cells, and right at this moment when I'm talking about it, when you're listening about it, there are millions of cells in our body that is undergoing cell cycle, the process of cell division. Say for example skin cell, cells in bone marrow, cells in the lining of intestinal, etc. So cell cycle, if I want to say what exactly is cell cycle is, it's an event, series of cyclic events, cyclic changes that a cell undergoes in order to divide. Okay, the cell needs to divide. For that it has to undergo series of changes, cyclic events, and it needs to divide. So that whole process of preparation of cell division, okay, this is how I remember, this is how I understand it. The preparation that it needs for a cell to divide and the actual process of cell division. Okay, these two things makes a cell cycle. Okay, more you will understand as we go on. Now it's a cell cycle. That means all the changes that are happening, it's in cyclic manner. It's not a linear pathway, rather it's a cyclic event. Wherever it starts, it ends at that point, and cell can again enter the same cycle once again. So it's a cell cycle. Again and again this can go on and on. So cell cycle, easily to remember, whatever series of events, cyclic changes a cell undergoes in order to prepare for cell division, and the actual process of division where everything in the cell divides into two. So one cell gets divided into two daughter cells, right? Talking about somatic cell. Now cell cycle, cell division is there in prokaryotes and eukaryotes both. It is quite different. We are talking about only in eukaryotes, and everything that I'm going to talk here is with reference to human somatic cells. So I'll be talking about mitosis everywhere. Alright, so let's start with the process of cell cycle. What are the steps and stages involved in the cell cycle? So here I can see that cell cycle has two major phases, right? Two stages are there. One where it has to prepare itself, because let's say for example this is one cell which has undergone division and it has produced two new daughter cells. So it's a newly produced daughter cell which is a small cell, and now it has to undergo next round of cell division. So it needs to grow first, right? It needs to prepare itself. It has just formed now. So it needs to grow, it needs to accumulate, prepare all the proteins, prepare itself for the division, right? So preparation, a lot of growing and preparation is needed before it actually divides. So that's why cell cycle is divided into two major parts. The first part would be the preparation of division, which is called interphase. Now it is called interphase because inter meaning between, right? It is a phase that is between two cell division, right? Because preparation is needed. No, again it has to divide. So before that it will prepare. It has just divided, prepare itself, divide again. So it is a phase that is between two divisions. So it is called interphase. And the second phase is actually when the division occurs, actually when the cell divides, which is called M phase. M stands for mitosis. So it is mitotic phase where the cell actually divides. Alright, so let's say for example this is a new cell that has just formed and it enters the next cell cycle. Okay, so as I just said, this is a newly cell that has just produced. Okay, so it will be a small cell. Now this cell, before it divides, it needs to grow. It has to prepare itself for the next division. So what it will do is, it will have the first phase of cell cycle. It will be in the first phase of cell cycle, that is the interphase. Now this interphase is again subdivided into three categories, and you will see why three categories and what are the use of it. The first phase subdivision of interphase is G1 phase, and here G stands for gap. Okay, it's gap 1 phase. The second phase is S phase. Okay, S here stands for synthesis. What each of these are, will come to it, okay, in just a moment. And the third phase of interphase is G2 phase, and G2 again here stands for gap 2. Now you know that, when cell undergoes a cell division, it is going to replicate its DNA. So synthesis of DNA will be occurring. That is obvious to understand. It is in the synthesis phase, S phase of an interphase. So S phase is the phase where the DNA replication takes place. Alright, so DNA synthesis occurs in S phase. Let me write it down. Alright, now G1 and G2 both are actually growth phase. Right, cell needs to grow, as I said, and prepare. So both of them are actually growth phase. So a lot of synthesis of proteins, RNA, whatever is needed for a cell to divide will be synthesized. Now G here does not stands for growth. Okay, it stands for gap only. And what happens in G1 and G2 is actually preparation of whatever is the phase followed by. Let's say, for example, G1 is followed by synthesis phase. In synthesis phase, as I said, DNA synthesis occurs, right? So for DNA synthesis, to replicate the DNA, cell needs a lot of things, right? Cell be needing proteins, so a lot of RNA synthesis, a lot of protein synthesis, a lot of nutrient accumulation has to be there, right? So in G1 phase, what all happens? First of all, cell needs to grow. So growth will occur. Cell needs to have all the nutrients. That cell has to be of adequate size. Okay, cell should grow of enough size. Then there will be a lot of protein synthesis, because DNA synthesis will require a lot of proteins. Proteins are needed, so of course a lot of RNA synthesis will be there. And it's, of course, you need energy, so a lot of synthesis of ATP also will be there. Alright, so whatever is needed for this synthesis, this G1 will take care of it. It will grow, it will accumulate all the proteins, everything that is needed for DNA replication. Cell is very metabolically active, so it will grow in size. Okay, so if the cell has entered over here, maybe this much it was when it entered, it will grow a little bit. Okay, it will be bigger in size, larger in size, because it's growing, right, and accumulating everything which is needed for the S phase. So now G1 will enter into S phase. G1 will be followed by synthesis phase. In synthesis phase, now the cell is dividing, right? Now cell needs to give rise to two new daughter cells. So it needs, duplicated DNA. It needs all the DNA to be replicated so that it can give both the daughter cell one copy of the DNA. So DNA replication, we all know it happens in interphase, specifically in the S phase. So the whole DNA will be duplicated, and along with DNA synthesis, also centrosomes would be replicated. Because finally both the copies of chromosome needs to be separated, right, by spindle fibers. So there has to be attachment to the chromosome. So centrosomes also will be replicated in the S phase along with DNA synthesis. So now cell has duplicated all the chromosomes. So what would happen is, let's draw it. So what happens is, in this G1 phase, for us, we have 46 chromosomes, or I would say 23 pairs of chromosome. So when cell is there in the G1 phase, we have 23 pairs of chromosome, right? So there are 23 pairs of chromosome, or 46 chromosome. Okay, so 2n is 46 in G1 phase, right? What happened is, when it came to S phase, all of these chromosomes duplicated, right? It replicated itself, made copy, So all the chromosomes made copy of itself. Alright, so what happens is 46 chromosomes are now doubled. Now when I say double, what do I mean? Look at this. Say for example, this is my one chromosome. This one chromosome have now doubled. What will I call this? This is one chromosome only, but there are two sister chromatids now. We have talked about chromosome, chromatids, chromatin, everything. So now these are duplicated chromosomes. Okay, if it is chromosome number 1, this is only chromosome number 1, but having two sister chromatids. And now why am I emphasizing this point is because I've seen students getting confused over this, saying that 2n would become 4n when it comes from G1 to S phase, which is actually wrong. Okay, chromosome number doesn't change. We have 46 chromosome. It is going to be 46 chromosome only, but in duplicated version. It is just copies that are attached. So 46 are 46 only. What changes is the content of DNA, the amount of DNA. Okay, make this clear in your mind. It is the amount of DNA that is increasing, not the number of chromosomes. So if my content here, if I say this G1 was 2C, that becomes here 4C, not 2n to 4n. Saying that would be wrong actually. It is the same number of chromosome but doubled. That's all. It is just replicated. One chromosome is now two sister chromatids. So in S phase, the content becomes 4C. Alright, now let's move on. Once the DNA synthesis has occurred, cell will move in the next phase. Now what happens is, DNA is double. What you need to do is now separate them and send them to two daughter cells. But separation, cell division itself is a complex process. So again, cell would need to prepare for division. Okay, so G2 is a phase, gap 2 is a phase where it prepares the cell for the division. Now what all we need for cell division would be prepared over here. Say for example, again we need a lot of protein. So protein accumulation will be there. You need ATP, of course, ATP synthesis will be there. You need organelles, right? Because both the daughter cells should have whatever is there in cytoplasm also. So organelle synthesis, replication will occur in G2. Now you can see here protein synthesis is there in G1 also and G2 also. So is there a difference? So what happens is G1 will prepare protein that are needed for DNA synthesis. The best example would be over here histone proteins. And in G2 is going to be followed by mitosis, right? The division, actual division. So in division you'll be needing proteins that are there in plasma membrane. So best example here would be proteins of plasma membrane. So there will be difference between the type of proteins produced in G1 as well as G2. So now G2 will prepare the cell for actual division. So now this whole thing, G1 followed by S followed by G2 is our interphase. Right, this is the phase where the cell is preparing for the division. Okay, and here the small portion what you can see, this is the actual division phase or M phase, where the mitosis occurs, cell divides, all the chromosomes get separated and go into two new daughter cell. Alright, so in M phase, again as you can see I have divided into two parts. We know that M phase or mitosis, all the chromosomes will be separated, it will go into nuclei, right? There will be separation of chromosome and two nuclei will be formed. So there is a phase of karyokinesis. Karyon means nuclei, right? So chromosome separation, that would occur over here. I would write it as mitosis. So this is the mitosis phase where chromosome would separate, and it will be followed by, of course, cytoplasm also needs to be separated and go into new cell. So followed by cytokinesis. So this is cytokinesis. I hope you can read it. Alright, so mitosis, we know again there will be different phases: prophase, metaphase, anaphase and telophase, followed by cytokinesis, separation of cytoplasm, and finally the cell will be divided into two new cells. Alright, now the actual cell division is taking only this much part. You can see out of the whole cell cycle, it is only this much part. If I'm talking about a typical mammalian cell, it may be only hardly 1 hour of a process compared to, the whole cell cycle, maybe taking 24 hours in cell culture, let's say, for example. So you can see a lot, majority of part cell spends in interphase, only the preparation, right? So I correlate this with the exam preparation that we do, right? Let's say, for example, you just started your semester. It will go for maybe 6 months, right? You prepare all the 6 months, prepare and prepare and prepare for the final exam, which is only of 3 hours, isn't it? So you prepare a lot, okay? All 6 months you are preparing to write everything only in 3 hours. For that 3 hours you give 6 months of preparation, sometimes even more than that. So that's just, something similar. The division process is so complex, it's so, a lot of things are happening that cell needs to prepare a lot for that. So majority of the cell cycle is actually the preparation. That's what we also do, isn't it? And the actual division is only a small part of it. So we started here, the cell went into a cell cycle and 2 daughter cells came. This cell again can enter the cell cycle, go in the G1 phase, or it will exit the cell cycle, come out of the cell cycle and enter something called as G0 phase or resting phase. It is not necessary that the cell will undergo a cell cycle and divide. So there are certain cells which will enter the resting phase and will be like that permanently, or it can again re-enter the cell cycle in G1 phase wherever it receives a signal. So let's take some example for that. So the first example is, once the cell finishes the cell cycle, it will keep on dividing, G1 to S, G2m phase, again it will enter G1 and continue the same thing. So these are the types of cell which undergoes the cell cycle again and again. So you can imagine cells which are exposed to wear and tear. Let's say, for example, skin cells. Best example, you have like we shed of skin cells every day, lots and lots of skin cells, so they need to be replenished, right? So skin cells would undergo cell cycle again and again. That's one example. So epithelial cells, lining of intestine, stomach, these cells would undergo cell cycle again and again. Okay. Second example is the cells, once they finish this cycle, they can enter into resting phase, and whenever a signal is given to them, whenever it's needed, they can enter back the cell cycle again, right? So such cells, let's say, for example, hepatocytes, liver cells, liver can regenerate if needed. So hepatocytes is one example which can enter in G0, and when proper signal is given, it will come back to G1 phase. And the third example would be some cells, when they are produced, they would enter G0 phase and will stop dividing at all. They are very specialized cells. Once they are made, they are going to do their function. Okay, that's what they will do in G0 phase. That's all. So such examples are neurons, skeletal muscles, specialized cells only to do their function. We know neurons, very difficult, cannot regenerate. It will just do its function. So based on the type of cell it is, it depends on, whether it will enter the next cell cycle or not, or G0 phase, how long it will be in G0 phase. Now, you see, a lot of things are happening, isn't it? A lot of things cells are dealing with. Here, still, we are just having an overview. There is much more in detail to this. So there has to be something which is actually making sure that everything is running properly and that there is no mistakes, because you can't afford having any damage or any errors in any of the stage, because otherwise your new cells would be defective. right? So there has to be some checking system, right, which is making sure that everything is fine and that you can go ahead and divide. So there are 3 checkpoints. Again, I'm just going to give you overview about it. 3 checkpoints in the cell cycle, during the cell cycle, which is making sure that everything is perfectly fine. So the first checkpoint is towards the end of G1. The second checkpoint will be end of G2. And the third checkpoint will be in M phase, to be very specific, in the metaphase. Now you might wonder why there is no checkpoint after S phase. So cell is very smart. What it does is, S phase depends on G1. So we have to make sure that everything is proper in G1, otherwise S will be affected. So there is a checkpoint. G2 doesn't depend on S actually, right? G2 is actually preparing for the division phase. So even if there is no checkpoint over here, that's okay, because G2 is not going to rely on S phase. But the division phase depends on G2 and S both. So at one shot only it will make sure that G2 and S has worked properly. So there is no checkpoint over here, saving the energy, direct checkpoint at the end of G2, and there is a checkpoint at metaphase, the exactly where all the chromosomes are aligned and ready to be separated, attached properly to spindle fiber. So that's a proper time to check whether everything is alright or not. Okay. So now let's just briefly have a look what these checkpoint does. So the first checkpoint is at G1s transition, right, where the cell is entering the S phase. So what G1 phase is doing, that is what it has to check. G1 phase, as I said, all the growth nutrients has to be produced, cell needs to grow in size. So it will make sure that cell has grown properly, the size has maintained properly, it has accumulated all the nutrients enough for the S phase. There is sufficient ATP, proteins accumulated needed for DNA replication, right? So G1 will make sure that the cell size is proper, all the nutrients are adequate, all the growth factors are there, the protein accumulation is there, ATP is there, all that it has to check. Only when it finds that everything is okay, then only it will allow the cell to enter the S phase. The next checkpoint is G2m transition, right? So by then DNA has replicated, a lot of other proteins, organelles have formed. So what this checkpoint would make sure is there any DNA damage, DNA is replicated properly or not? So it needs to check for that. It will check all the organelles are ready or not, because you need everything to go into new cell. So it has to be, everything has to be enough for both the cell. There has to be again ATP synthesis, protein synthesis. Only when all these things are done, cell will be allowed to move in the M phase. And then the next or the last checkpoint is in the M phase. To be very specific, it's in the metaphase. So this is called Spindle Checkpoint, where the important thing cell does is to check whether all the chromosomes are attached properly to spindle fiber or not, because both these copies needs to go into new cells. So it has to be attached at both the side for separation. Also, all the chromosomes should be at equatorial plane, right, in metaphase, we know that, so that it gets pulled both the sides. So it will make sure that all the chromosomes are attached and lined on the equatorial plane. That's what is the work of third checkpoint. Once it is done, it will be allowed to move in the next phase. Now, if there is any problem in any of these checkpoint, it is found that there is any problem, either the cell will go back, repair itself and restart the whole thing again, because you can't afford having any error, or it can also just remove the cell from the cell cycle and just discard the cell. Okay, it will not be allowed to undergo the cell cycle. So that's the importance of checkpoints. Now here I talked about this content, right? So in S phase it was 4C. In G2 again it is 4C only because it is still double. What happens in M phase? The chromosomes will divide. So at the end, when two new cells are produced, again the content is 2C. Of course it is 2N only because we are talking about mitosis, and the content is 2C. Alright, that's just additional information. So that's it. That's pretty much all. As an overview, you should know about cell cycle. So that's all for now. I hope this video was helpful. Do subscribe to the channel for new video every week, and I'll see you next time. Until then, keep learning.